Welcome to Discover ASAP, the video interview series where we highlight the top findings and tools connected to the ASAP network. Today, I'm talking with Miguel Gonzalez-Lozano, a postdoctoral fellow on Team Harper, about their EndoMAP tool. Miguel, thank you so much for joining me today. Can you briefly start off by telling us about your ASAP project and the impact you hope to have on Parkinson's disease?
Yes, of course. Thank you for having me. So, EndoMAP is a project in which we aim to understand the molecular architecture of the endosomal system. And, we decided to go to start studying the endosomal system because in general, the endolysosomal system has emerged as a major vulnerability risk for neurodegeneration in general, for Parkinson, but also other diseases like Alzheimer's disease. So, one of the things that there's many risk alleles or genes that have been identified in patients that are either proteins that are resident in the endolysosomal system or that they interact with the system and these organelles. This project came with one of the problems that we have encountered in this field is that how do we get farther from this identification? There's, as I mentioned, there's a lot of genetic studies that they have pinpoint on specific proteins that are located in this system. But it often happens, and is very frustrating for us researchers, that whenever you find a gene that is involved in something important, and then you look at, okay, what do we know about this protein? And there's not so much known about it. So then the question is like, how do we get farther? It also can happen that, maybe a lot about your protein, but you just don't know how it's integrated functionally in the system or connected. So it's very difficult to interpret what are the consequences of having a specific mutation or a specific impairment in this gene. So then how do we get farther and how do we actually end up fixing it? And this is kind of the inspiration that brought us to EndoMAP, and is that we wanted to prevent to have this dead end in the research because we just don't have enough knowledge to go the next step. So the aim is really simple. It's just like we wanted to gain knowledge on the endolysosomal system. So we really took a very mechanistic approach or view in which, like we said, well, let's take the system and let's try to dissect all the pieces and how do they assemble together, so then that we can know much better whenever we detect like this is the piece that is missing or there's a defect in here, we are going to know much better how to fix it and how to replace it. So the end of this project is really try to understand how is the proteome landscape of the endosomal system.
Great. Yeah, I think this is true that there's often these kinds of roadblocks in discovery in Parkinson's research. There's so many unknown questions, and so it's important to have these kinds of projects that help sort of create resources for researchers to help bring those barriers down. So, as you've mentioned, this tool that you guys have created is called EndoMAP. So, can you give us a brief overview of exactly what it is and the kinds of data that people can explore in EndoMAP?
Yes. So, the name just come from the idea of that we are going to make a map of the endosomal proteome. So, as you mentioned, this is a resource, and the idea is that we wanted a resource in which we can be able to mechanistically understand how the proteins in the endosome compartment works. So what we wanted to then know is what are the proteins that are there, how do they interact with each other, and how are they assembled into structures and in protein complexes. And we wanted to do this for as many proteins as we can from the endosome. So we started simply I started just by looking at characterizing the proteome itself, so which proteins are there. So I basically took almost all experimental data that is publicly available in the last 10 to 15 years, and also some of my experiments, put everything together to make like a comprehensive list of proteins that are known to be there, but also proteins that are known to be there, but that we don't know yet what they do there. And then to further characterize those, then I first purified endosomes using EndoIP, which is a method to purify these organelles. And then I look at the contacts and their interactions by using two methods that are proteomic methods based on mass spectrometry. So one is cross-linking mass spectrometry and the other is cofractionation. So in cross-linking, what I use is small chemicals that are going to bind to proteins, and they will bind specifically they are going to connect points of proteins that are very close by in spatial distance. So we can identify these connection points, so we can know what are the interactions and in which regions they occur. And then with cofractionation, we are going to extract all the protein complexes still in native state, and we are going to separate them so we can identify the components that are in each of the complexes. So by that we identified interactions, and we have this is done in large scale, so we have a large repertoire of interactions. And then what we did is we took all these interactions and we use AlphaFold to predict what is the structure of these proteins when they are interacting. And we start building from pairs of proteins, we start building bigger and bigger to have complexes that have more or higher order interactions. So we end up having this repertoire of also predictions of these proteins.
Great. That sounds really useful. So I'm curious, you've touched a little bit about the need for a resource like this, but I'm curious if there are other similar tools, or, what advantages does EndoMAP have over other tools like it, or is this really the first, of its kind?
I mean, it's a bit of the first, because there was some studies done in endosome in general, right? Like in which proteins are there. But there's not so many that are really dedicated to the endosome. And the reason is because it was very difficult to purify it before. So now with the EndoIP methods that have been developed in the Harper lab, we can much nicer analyze this compartment. And the type of data is completely new, because it's the first time that we have applied, this large-scale cross-linking mass spectrometry to the endosome, and also again the first time that we have done this large-scale AlphaFold screening. Like at least it's the first one that is really dedicated to the endosomal compartment. So there's two keys here that make this very new, is that this is untargeted. So we simply took the organelle and analyzed it whole. So to try to identify as much proteins as possible. So we are not targeting specific proteins, which means that we are also open to really new discoveries and things that maybe we were not expecting. And the second is that this is done we are studying these proteins here in their physiological environment, because we isolate the organelles and we try to identify as much as we can within the organelle, before extraction, etc. Sorry.
Yeah, that makes a lot of sense. I think that is a particularly, I think, exciting thing about EndoMAP, that it is really unbiased in the way that you've collected this data to, open the door to new discoveries. So, how do you think this tool contributes to the Parkinson's field, or how do you hope that it might contribute to the Parkinson's field?
Yeah, so I mean, as you mentioned, I think the best way that this can help is as a resource, and I think one of the keys is about accelerating the research. Because as I was mentioning before, I think this is really filling the knowledge gaps that we had on the endolysosomal system. So we can now bypass these dead ends that we sometimes encounter when we are doing research, and we just don't know how to get further. So I think this platform is really going to help with the interpretation of the findings that people have, and then it's probably going to give the researchers the clues that they need to generate an hypothesis and go to the next step, so they can continue trying to untangle what is the mechanism of the pathology, etc. And there's also another key that because of this revolution of AlphaFold, now we are looking at these proteins in 3D, and their structural features, which will help down the road, as a platform for helping in the drug discovery. And I actually, maybe I can give you an example that we have included in this publication that is about a protein that is called Team 230, which is a pretty enigmatic protein that it's not much is known. So it's one of these examples in which, there was a family that has familial Parkinson's disorder, and there was a few variants of genetic variants that were located in this protein, but there's not much information. What is it pathologically relevant? What is this protein doing? There was really not much known about it. So we took this as an example and looked in our EndoMAP how can we go further with this protein. So something that was very interesting is that we identified this as a new component of an ATPase flippase that controls the organization of the lipids within the endosome organelle. And we actually identified that these variants were located at the interface of the interaction. So if you disrupt these variants, then the interaction is lost. And we started to, following up a little bit on this protein by making several models on ES cells that we can differentiate to neurons, so we can see what are the consequences of these variants, in human neurons, etc. So this is one example of how we think this can help in the field of Parkinson.
Great. Yeah, that's super exciting. I love that you shared an example of in your own, in the Harper lab, how this has already started to really lead to new discoveries. So, lastly, and potentially most importantly here, I want to ask you how someone can access EndoMAP to use in their own research.
Yeah. So, well, all our data is open access. So we have, there's, as I mentioned, there's different types of data. So some are in data repositories. We have some part in Zenodo, reagents in the corresponding repository. So everything is accessible. But also to facilitate the access to the data, because it's relatively complicated, because it's so much and everything needs to be integrated. We created a web platform. And then maybe actually I can go ahead and share my screen. So this was done by Ernst Smith, which is in the Johannes Walter Lab, and he really kindly prepared this for us. So we generated this website in which all data is there. So we decided not to hide anything, because we understand that maybe some researchers have a different perspective of the data or want to look into it, in a different direction. So we decided to put everything, which means that there's things that also maybe have the evidence is not so high. So I just wanted to ask people to be critical about what is here. So here is the interaction network. In each of the nodes represents a protein, each of the edges represents an interaction, and then you can either click on them or you can also search. So for example, if we go to this protein that I was mentioning before, Team 230, we will go to the dedicated network. So here you can click on the protein and you can see what information we have about it. You can click on the interaction and you will see the different types of evidence. So that support this interaction that we have found in EndoMAP, where if it's cross-linking or if it's co-fractionation, if it's something from before, maybe it was already known. So that's what I'm asking. Like the interactions, some of them are going to have way more evidence than others. So be critical and about if you want to follow up on something. And then if you click here, we go to a different page, which I hope that you still can see.
No,
actually,
I'm still on the page. Oh, there we go.
Here we go. So when you click there, you will go to that specific interaction. And if you scroll down, you can see the AlphaFold prediction and of the different proteins, so you can play around. You also have the information on the contacts that we identified by crosslink, etc. So with this you can explore your results and see if you generate the hypothesis to make the next step.
Great. That is wonderful, and I think it's super useful to be able to show people exactly what this resource that you have created looks like. It was great to hear about the work that you and Team Harper are doing to further Parkinson's research. So thank you so much for chatting with us.
Thank you.